everyone. Welcome. Thank you for being here. My name is Jain. I'm an HKS master's student here and the co-chair of India Caucus. I'm so happy to collaborate with IIT Agni. This is, I think, our first collaboration with IIT Agni here, and looking forward for so many more. So thank you all of you for making it possible. Now I'd invite Samya Mishra, who is an alum from HKS, and she is on the board of the IIT Agni executive board member. So I'll invite her to take the event away. Thank you, Samya.
Good afternoon, everyone, and thank you for your patience for the initial 5-10 minutes and the commotion that we had at the entry. I'm so glad to have you all here. On behalf of the IIT Association of Greater New England and the Harvard community of Indian students and alumni, it is my distinct honor to welcome you to today's event. It is an immense privilege to introduce our distinguished speaker, Dr. Rakesh Jain, an eminent scientist, visionary leader, and pioneer in the field of tumor biology. Dr. Jain's academic journey began at IIT Kanpur, where he earned his BTech in Chemical Engineering, class of 1972. He then pursued his MS and PhD at the University of Delaware, laying the foundation for a groundbreaking career that has transformed our understanding of cancer treatment. Currently, Dr. Jain serves as director of the Edwin L. Steele Laboratory for Tumor Biology at Massachusetts General Hospital. He's also the Andrew Cook Professor of Radiation Oncology at Harvard Medical School, among many other appointments that he's currently holding. His contributions to science and medicine have been globally recognized. In 2013, President Barack Obama awarded him the National Medal of Science for his revolutionary work in tumor research. He holds the extraordinary distinction of being a member of all 3 national academies of sciences, engineering, and medicine. With over 800 publications cited more than 100,000 times, he ranks among the top 1% of cited researchers in clinical medicine. He has mentored over 200 doctoral and postdoctoral scholars, shaping the future of biomedical innovation. Dr. Jain's research has uncovered critical physical and physiological barriers in tumors that limit the effectiveness of modern therapeutics. His pioneering work in molecular, nano, and immuno-therapeutics has led to groundbreaking strategies, many of which have been successfully translated from bench to bedside through multi-disciplinary clinical trials. In today's talk, Dr. Jain will share his profound insights into tumor biology, cutting-edge imaging technologies, and how his research is shaping the future of cancer treatment and even other diseases like tuberculosis. With that, please join me in welcoming Dr. Rakesh Jain with a round of warm applause. Thank you.
Thank you very much, Samya, for such kind words for introduction. And thank you very much for inviting me to IIT Agni. I had the opportunity to meet some of you at the Pan IIT meeting early last year in Washington, D.C. And it's always a pleasure to come back and see IITians and other fellow Indians. So I'm going to make this very informal. So please interrupt me, ask me questions. Since I'm a professor, at the end of this whole talk, there'll be a quiz. And so be prepared for that, okay? So I came to this country in 1972, as Samya said, after finishing my bachelor's in chemical engineering from IIT Kanpur. And I came here because I worked to work with a very distinguished professor of chemical engineering at Delaware, Professor Morton Dan. I had used several of his books, textbooks, in my undergraduate at IIT. And his PhD student was a faculty member at IIT Kanpur. So he convinced me that I should go to Delaware and work with him. And Delaware was at that time the #1 school of chemical engineering, courtesy primarily of DuPont's that had endowed so many chairs there. the richest department at that time. And I had a very good fortune working with it. And so for my master's thesis, I worked on mathematical modeling of Delaware River, especially of how to control pollution in the river. And wrote about it. None of you probably know in those days there were no computers like this. So you have to type cards, punch cards. And so my PhD, my master's thesis had a 2,000 card program. So you should carry in a big box like this, submit it overnight, and next day you would get the results. And then you make changes and modify things. And so anyway, fortunately, I finished my PhD in March of 1974. My master's in March of 1974. And then Professor Dan said to me, why don't you just, you've done so much in your master's, if you just spend another year, you would easily get your PhD in a very short time. That sounded very tempting. Just finishing up your PhD in about 2.5 years afterwards. But at that time, Indians had the reputation of being very good in mathematics, but not being able to do anything with their hands in the lab. And I still am not very good with my hands in the lab. But I said, you know what? This is the time let me find out about it. So I started shopping around for PhD project. And I had the good fortune of meeting Professor James Way at Delaware, who had joined, oh, can you hear me?
Not so well.
I think the mic could be a little closer to him. Yeah. I mean, we can, but probably not that.
Can you hear me now?
Yeah. Maybe we adjust it a little bit.
Thanks for...
Could you raise the mic a bit? Yeah.
Only my wife would tell the truth. It's Dr. Jackie Sampson, who's also a faculty member at Harvard Med School. Yeah. So do you want me to repeat everything?
That's up to you.
Do you want me to repeat everything?
No.
How about in the last row? Could you hear me? Okay, all right. It'll be part of the quiz, okay? So, Professor Way had just joined University of Delaware. He was a director of research at Mobile Research Corporation. And he came and he said to me, do you want to work on cancer research? And I said, what? I'm in chemical engineering department. He said, do you want to work in cancer research? I said, on what? So on looking at drug delivery in tumors. That's a chemical engineering problem, he said. But I said, I don't know any biology. Because if you remember in India at that time, and maybe now, in high school you have to choose if you're going to be in the medical direction, then you would go to biology, or not. Engineering, you would go to math. Besides, I went to my first biology class, and I'm a Jain, and they cut open a frog, and I just fainted. So I said, okay, this is not my cup of tea, you know. So I dropped in biology and took Sanskrit in 9th grade. And I had not taken Sanskrit in the 6th, 7th, and 8th grade, so I had to learn the first 3 years Sanskrit, and nevertheless I did that. And so I told him that, I don't know any biology. He says, don't worry about it. So he gave me a textbook of human physiology, which is used in, which we use at Harvard and other places for medical students. And he said, see, if you read this, it makes sense to you. I read it, and I went back to him and said, makes sense. Yeah. So he said, okay. So then he drove me to NIH, which was very close to Washington, to University of Delaware, like within 2-hour drive. And he introduced me to a very distinguished cancer biologist, pathologist, Pietro Gallino. Pietro was an eminent tumor biologist, pathologist. He was the head of breast cancer for the whole country, the US research. And Pietro had developed a tumor in rats which had a single artery and a single vein. Now, just as a background, when tumors grow in our body, they get blood supply from many places. But if you have a tumor with a single artery and a single vein, you can inject something in the artery and measure in the vein, and from that you can find out how much is what's being picked up by the tumor. And he was using that to look at how much oxygen tumors consume, how much glucose they consume, how much lactic acid they produce, etc. So when he showed it to me, I said, has anybody used this to look for how much drugs are taken up by tumors? So in the beginning he said, yeah, that's not an important problem. I said, why not? He said, well, because that's why they give drugs to people. They obviously get into cancer. Why would they not give it? I said, but has anybody looked at it? He said, it's not an important problem. But I said, I'm a chemical engineer. It would be a good engineering problem for me. So I looked at Professor Wei. He said, yeah. So I said, you know, I'll look at how much drug, at that time most widely used drug in cancer was methotrexate, is going in artery, and how much comes out the vein, from that figure out, and then at the end cut the tumor and see where the drug is. So that sounds good, but he said, how are you going to get a chemical engineering degree out of that? And I said, well, I'll do mathematical modeling. I said, that's what engineers do. And they do input, output, and from that try to get a sense of what's going on in a black box. He said, yeah, that sounds good. So they both agreed, and I said, okay. Now notice that I'm a Jain. I'd never touched a mouse or a rat in my life. But Pietro had a very talented technician. She'd been working with him for nearly 30 years. So she says, so he introduced me to her, Flora Grantham name. He said, look, we'll do the experiments together. You do the collection, measure. And so I used to go every Sunday night, I'll drive to Washington, D.C. Monday morning, I'll start the experiments. Friday night, I'll take all the data, go back to Delaware, analyze it over the weekend, go back Monday. And essentially, the big finding, for me, the discovery was that not much drug was getting into the tumor. Most of the drug was just like bypassing the tumor like a highway around a beltway around the city. And when I cut open the tumor, most of the drug was sitting in the margin. I said, how is this going to work? And actually, the drugs weren't working very well in tumor, even though they were being given to patients. They are not very effective. So, so that's really got me started. And I finished my PhD in December of '75. And December 8th is when I defended. And in January, I was an assistant professor at Columbia University, New York, in chemical engineering. And looking at me now, you would not realize I had hair, but I had my students who are teaching, some of them were older than me, so I said, how are they going to take me seriously? So I decided to grow a beard and just begin to look very, I wish I had all those old pictures. And so I could look, they would take me seriously then. Anyway, the long story short, Columbia was not pleased by my doing cancer research. They wanted me to do more traditional chemical engineering, even though I was teaching traditional courses. So then I moved from there to Carnegie Mellon University in Pittsburgh, and there I was welcomed by the president of the university, and they had given me facility to do the animal work in chemical engineering department. And that's where my journey began. And what I'm going to share with you is the journey. And essentially, so the question I wanted to ask is, why don't the drugs get into the tumor? And how can we improve the delivery? So to understand that, let me just give you how drugs get into the tumor. Let me give you a lecture on Cancer 101. So when we think of cancer, we usually think of cancer as when you say somebody has a breast cancer, we think of just a collection of breast cancer cells. When you think of pancreatic cancer, you think collection. Did you know that the pancreatic cancer, only 5% of the cells are cancer cells? Did you know that? And that is the most difficult tumor to treat. The most difficult tumor to treat. And you know what is the easiest tumor to treat? A leukemia. Those are 100% cancer cells. So that tells you what we need to worry about is not cancer cells. Everybody's worrying about them. Let's worry about what's around them and what is it stopping them. So if you look at a tumor, say breast cancer, these dark cells, these are the cancer cells. And these cells are nourished by nutrients for these cells comes from the blood vessels. All right? And then the drugs and nutrients come in, cross here. But these cells are embedded in something known as the extracellular matrix made of collagen and hyaluronic acid. But in addition to the cancer cell, there are a variety of normal cells in the tumor, and known as host cells. And these include fibroblasts. You don't know all the names and everything. A variety of immune cells that are either stationary or come and go. And all these are nourished by the blood vessels. And the excess fluid, excess material is drained by what is known as lymphatic, shown here in the green color. So you can imagine if blood vessels are not functional, you don't want to deliver drugs. Or even if they're functional, if they can't get out of the vessel wall, they're not going to work. Or if they get out and this matrix stops them, they can. All right? Actually, the reason pancreatic cancer cure has not increased for nearly 20, 30 years is because it's got too much matrix in it. 90% of the tumors is chock full of matrix and fibroblasts. So the question I asked is, to understand this, what we do. So I wrote my first grant to NIH, and that got rejected. By comments like, what's a chemical engineer doing? Cancer research, etc., etc. And so I didn't have money, so I did a lot of my initial work by doing mathematical modeling. You don't need much for that. You need a paper and pencil. And you don't always support students. You can do it yourself. Right? But mathematical modeling can do only so much. You can make all kinds of predictions. But can you hear me, guys?
Yeah.
Can you do something about
A little louder.
I think maybe let's try that. That maybe
Let's
that's
do
all.
I'm so sorry. Did you guys hear any of that, or you just said, okay?
We are able to hear you.
Yes, clear.
No, little louder.
If you can even move to the front, right?
Yeah.
So those who cannot hear, why don't you move to this corner here?
The seats will also.
I mean, this is a small room. I can come in the middle and. Well, let's see. No, I don't know. All right, guys, can you hear me now?
Much better.
Very
Is
good.
this working?
Yeah.
Is it a volume control? I can crank it up.
It should say control. I can control it from here.
All right, could you please? Yeah.
Maybe you can move the lapel a little higher.
I'm a pretty loud guy.
Yeah.
Oh my
Now-
God.
now, is
Yeah.
that- now.
So,
Yes.
is this too much? Can you?
No, this is good.
Can you?
Keep it there.
Are you going to repeat the whole thing now?
Yeah, I'm going to start. So I came to this country in 1972. And so I did mathematical modeling. As I said, with mathematical modeling, you can make all these predictions and all. But they have to be vetted with the reality, right? You need to be able to check your predictions. And so, how do you do that? And at that time, the only way one could do these things, when I entered the field, was the tumor was like a black box. You could just do imaging, but that'll give you one time point at it. It would not give you the dynamics. Or if you do CT or MRI, the imaging, the resolution is very poor. So what I did is I decided to put glass windows in mice. These guys came just at the right time. And these glass windows, you can put anywhere in mice, in the brain, to look at brain tumors or brain metastasis, breast, to look at breast cancer, liver, and so on and so forth. This is a window on the back skin. It's known as dorsal skin. The beauty of this is now you can put this mouse in a microscope and you can see in real time what's going on at a very high resolution. And then, so we did this and we got a lot of insight from that, but that was not enough for me. What I wanted to do was to check if the predictions, what we are seeing here in animals, also hold up in cancer patients. So I've been very lucky in my life that I've had very good clinical collaborators. So we have been able to do about 40 clinical trials in my 20 plus years, where we have taken findings from here and brought them from bench to bedside. And then from these we have taken lessons and went back to, again, to the bench to see how we can improve the treatment further. So what I'm going to do is share this journey with you now. So when these windows are about 1 centimeter, and if you put about 100,000 cancer cells on day 0 here, see in day 5 you see cancer cells are growing. And you begin to see some changes are occurring in blood vessels. But this is at lower magnification, so you cannot see all the teeny vessels. But you can get a sense that the blood vessels are changing. Right? Yeah?
Sorry, Doctor.
Yeah.
So these windows are the surface, are they inside, like?
They're on the surface.
Surface.
Yeah. But we have microscopes which can look at deeper.
Yeah.
Not your regular, this is taken with a regular camera.
Yeah.
But we have microscopes.
Yeah, sure.
Which can look at deeper. But not only can you look at tumor growth, look at this, growth of the tumor, but you can look at regression too. So this is a hormone-dependent tumor, in this case androgen-dependent tumor. So if you perform orchiectomy, which is a medical word for castration, and you see that the hormone androgen is gone, and then you see there's a regression of the tumor, because it's the standard therapy for prostate cancer, right? And this leads, or not by castration, but by drugs, which essentially have to do. You see that the tumor regresses in every tumor. We give it therapy, the tumor regresses, and then it takes on. Sometimes you're lucky, this doesn't occur for years, decades, but it invariably occurs in tumors, except with immunotherapy. Yeah?
So, as the tumor is growing, there are blood vessels that get formed, right?
Yeah.
And so when the tumor recedes, do the blood vessels also stay there, the new blood vessels, or?
The vessels regress.
Okay.
Yeah. The vessels regress
So
first.
vessels will also regress.
Yeah, actually, again, since you're asking a detailed question, what we discovered in this paper was the vessels regress first before cancer cells die. You see, that's because the endothelial cells are more dependent on this hormone than cancer cells are. You see what I mean? So this was the discovery here. Yeah. Anyway, so as somebody had asked me, can we look at deeper? So this is the brain tumor growing in this quadrant of a mouse, and the vessels are color-coded, so you can see a different depth. Right? So what you see here is that, oops, this thing fell down. I don't need this, right?
What you do, maybe we just did it for you.
Let's hope it stays.
Yeah, this
Can
is
you hear me?
That's much better.
Yeah.
You guys need to raise your hand, you know, if you can't hear me. Yeah. No, that's
No, as long as it stays clipped.
well, if it falls on, please tell me, okay? I usually don't look at my lapel, yeah. All right, so these are all the blood vessels on the outer windows. What happens in colon cancer, interior? So for that, somebody while chatting asking me a question, do blood vessels change when a tumor is grown in different places? And the answer is yes. So if we take cancer cells from the same petri dish, and put them in 3 places. If you take breast cancer cells, and you put them in the breast of a mouse, put them on the brain metastasis, or put them in the back skin. And now you look at the tumor and these 3 different sites. Okay? Same cancer cells, 3 sites. What do you see? The vessels look very different. I forget who asked me that question. Earlier. Yeah, you did. So you see that? The vessels are very different. Yeah. And they function very differently. So for example, if you give a drug, this tumor responds beautifully. But this tumor doesn't. All right? This is refractory. The brain met doesn't respond. Skin responds. All right? So while these vessels look different, there's one thing common among all of them. They're all abnormal. Yeah?
So, just to understand, so for the breast cancer, you said the drug works beautifully there. Is it because of the aggregation of those blood vessels? So it needs more, like if it is sparse, then the drug cannot work well. Is that why it works?
No.
Or do we know the reason why it does?
Yes, we do. The reason is that these vessels are not very as permeable, number one. But bigger problem is not blood vessels. The bigger problem is that when cancer cells go to the brain, they begin to make things. They begin to tell the other cells, the host cells, to make molecules that stop the function of the drugs that work here. Yeah. I don't want to go into all the details of that, but if you keep writing the reference at the end, so you can refer the paper. Yeah, this was a major discovery because people who work in pharmaceutical company targeting a certain mutation, they think if they can have a drug that will target the mutation here, it'll also target the mutation here. It doesn't work. It kills the cells here very nicely, and this is where the failure of molecular medicine is, the targeted therapy. It was supposed to cure cancer originally because we thought, okay, once we know all the mutations, we can cure cancer. Do you know how many cancer patients benefit nowadays from targeted therapies? 5%. This was supposed to be because we understand the genetics. It's a joke. And the reason is they forgot about microenvironment. All right? That the microenvironment here confers resistance to what works here. But again, you guys are slowing me down. Are you prepared to stay till 4? Because I can go on and on. I'm really, this is my passion, okay? So I'm not worried about me, I'm worried about you because you might starve or fall off. Yeah, go ahead. Question, somebody would, yeah.
Can you explain what we see here? There's a yellow colored thing and the red colored thing. Is there a difference between them?
What?
There's a red colored cells and
No.
All of them.
All right, so we decide these are all outside tumors. What about inside? Tumors inside our gut and other places. So we developed an endoscope so we could do the same thing in, this is the colon of a mouse. And you see how these blood vessels for the colon are very nicely organized? Look at them, they're beautifully organized, right? And I'll just show you on fast view, like opening up. See that, how nice they are? And they're beautifully arranged so that every cell of the gut gets enough oxygen. All right? Now what we're going to do is work with a mouse that develops this tumor. It's a genetically engineered mouse. So this is an 11-week-old mouse, and you see the tumor beginning to arise here. See the blood vessels don't look beautiful like you saw that? Now wait a little longer. This is a 13-week-old mouse. See that? See what's happening? Now you wait a little longer, 16 weeks. Now look at this. It looks like somebody dropped a bomb here, right? You don't see any vessels here. How are you going to deliver a drug? So these are the things people are not thinking about when I entered the field. They still don't think about it. That's a sad thing. They still think, oh yeah, once you have figured out a mutation in a cell, you make a drug, it's going to cure it. And it's a joke. And so, I've been saying this to people who do this, and I give talks in front of them, and you can imagine I'm not very popular with that. Okay, but this is, you can't stay away from reality. This is what it is. And so once you show this in a talk, they say, okay, well, this is all Mickey Mouse. Does this happen in patients? All right? So I've been very fortunate. I've had very good surgeons as my postdoctoral fellow. So I had a surgeon, Mitch Posner, who's right now chief of surgery at University of Chicago Medical School. And he was a postdoc in my laboratory at Carnegie Mellon University. And what Mitch did was, this is a 0.5 kilogram colon cancer from a patient. And just before he was about to resect the tumor, what he did is he injected a monomer into the artery of this tumor. And then once the monomer came out of the other side, right, then he put a catalyst. And then the catalyst came back like this and it polymerized it. So now suddenly, everywhere there were blood vessels was full of. I mean, he didn't do this inside the patient, obviously. He took it immediately after resection, he did that. And then what he did is he took this 0.5 kilogram tumor and put it in KO8 solution for a month. So he ate away all the things except which were not polymer. And see, then we took photographs, high image resolution. You see that? The vessels here are screwed up as I showed you, right? And they're avascular regions which are no blood vessels, similar to what I showed you. So what I was showing in Mickey Mouse is representing what's happening. This has been my guiding principle my whole career. Check things in patients so that you're grounded in reality. Right? So, but what's common among all this is the following. These are the structural abnormalities. But what is real is structural abnormality is bad enough. What is really bad is the functional abnormality. So now, you look at the flow in real time. This is something at very high resolution you cannot do with any other technology. What you see is the blood flow is pretty good in some regions of the tumor, quite brisk in some of these vessels. But you go nearby, a blood supply. Now, how are you going to deliver your magic bullet? You can't. And what is worse, as our work showed over the years, is this region would not have enough oxygen. Now, you would say, okay, enough oxygen, when the cancer cells die, they would not die. Cancer cells can live on garbage, okay? So what happens is that these cells are protected cancer cells. So if you take these cancer cells and put them somewhere else, they are more metastatic, they're more aggressive than these cancer cells. So if you cannot kill these cancer cells with your drug, you can see what's going to happen. Relapse will occur. So when I saw this in real time, the question became, what do we do about it? How do we fix this? And it took me quite some time to figure out how to fix it. And let me show you, but I was thinking about how to fix it before I knew how to do it. So I, as a chemical engineer, wrote, when I was a professor of chemical engineering, oh, by the way, as I said to you, when you don't have blood supply, you hypoxia, then you low pH, low oxygen. And normally blood vessels are abnormal in tumors, so are the lymphatic. Remember I showed you the draining vessels? And they had never been imaged before in any animal. So I had a postdoctoral fellow, a doctor from Switzerland, Anders Lohr. He did the very first imaging of these, but he injected 2 million molecular weight dextran in the tail of a mouse. You see these chicken wire meshes? Those are lymphatics. They look like, these are lymphatic capillaries. Now, in the same tail, if you put a sarcoma, a solid tumor, right here, do the same imaging, can't see any green color. Lymphatics are not functional. They may be there, but they are not doing their job. And it has implications. And by the peri-tumor lymphatic, the outside, they are bigger. They can, the word for medical word for that become hyperplastic because they have to drain all this fluid. So just imagine, if you have this room and you keep filling it up with water, what will happen? Pressure will begin to go up. That's exactly what happens in tumors. The hydrostatic pressure goes up. We're the first people to report that, and why, and what its implications are. So first we again measured in hundreds of mice. Same joke, this is Mickey Mouse work. So then we made measurements in patients and look in the breast of a woman or a normal skin, the pressure is around 0 millimeter of mercury, but in every solid tumor it's much higher. Our mean arterial pressure is about 120. Look at this, it goes up to 40. All right? So now you can imagine when the fluid pressure is so high, it pushes drugs out from the tumor to the margin, to the surrounding tumor. So that's another reason why drugs get pushed out from the tumor. So essentially, to summarize this work of nearly 2 decades, what we showed here is that if you have abnormal vessels, you have low oxygen, acidity, low pH, high fluid pressure, and that confers resistance and facilitates tumor growth and metastasis through 8 different mechanisms. I'll not go into the biology of that, that's a little too much. Happy to talk about that, or you can look at this review article, which is about 11 years old. And so now what it tells you, there are 8 problems it created, 8 category of problems created by this abnormal vessels. Now suppose you develop drugs, and to solve each problem it costs $100,000 a year per drug. That's a pretty conservative number. You need $800,000 to solve this problem created by blood vessels, right? So now I want you to think about the analogy. You're standing by Harvard Bridge, right? By standing by the river, and you're seeing these corpses go by, right? And say, what's going on? Why? So you can take the corpse, and if you're a pathologist, you analyze, okay, what's the cause of death? Or if you're an engineer like me, you'll walk up the river and ask the question, who's throwing these guys in the water? All right? And so we say, okay, let's find out. Let's just work up the river, see how we can, why is this blood supply poor, and how can we fix it? Are you with me so far? Yeah. So how do we do it? So our overarching hypothesis, and again, this got too much jargon, and don't worry about it. Essentially what we're saying is that the blood vessels are abnormal, shown on the left-hand side. It creates a lot of trouble. It's highly immunosuppressive. But if you can improve the blood supply, vascular function, then we could create what we call vascular normalization. We would create an immunosupportive environment, drugs would get, all kinds of nice things would happen. So the challenge is how do you do it? So our laboratory has come up with 2 ideas. That's what we are known for. One is known as vascular normalization, where the idea is that, why is this cursor? The top one, the vascular normalization. That's what really our laboratory is famous for, this idea, where we are saying that what we need to use are drugs that can repair the blood vessels. Right? And what was interesting here is we proposed to use the drugs that were originally developed to destroy the blood vessels. So it was 180-degree thinking. All right? Totally counterintuitive. So you can imagine that resistance I got to that, and I'll talk. Second, as I said to you, that a lot of tumors like pancreatic cancer, which is incurable, is that there's a lot of matrix which compresses the blood vessels. All right? So even if you have a blood vessel that's squished, you can't have the flow. So if you can figure out somehow to deplete that, that would be good, right? So these are the 2 strategies our laboratory is known for. So before I knew any of these strategies, how to do this, in 1998, when I was a professor of chemical, I was a professor of chemical engineering at Carnegie Mellon, I wrote 3 review articles, and essentially putting the framework for how would you repair the blood vessels. So just imagine you're watering your lawn, and the water stops flowing. What do you think happened? Why would that happen? Leakiness?
Kink in the pipe.
Kink in the pipe, okay, yeah, it's a good point. Anything else?
There's no water flowing through over time.
Is what?
There's no source.
Yeah, there's no source.
Well, that means killing the heart, that's a little too much. You don't want to. You can, but that's not a way to cure cancer, right? Yeah, so
Plaque formation within the
kink, and what else? Occlusion, okay.
The pressure, interstitial hydrostatic pressure.
How about if your daughter decided to ride her bike? And that will do it too, right? But that's almost mathematically same as a kink, right? Since you reduce the cross-section area to 0, right? So the question is, if you can figure out the force applied by this matrix, and then figure out how to get rid of this, you will be able to start flow. All right, this was in '98. I didn't know how to do this. I did not know how to measure it. But again, when I came to MGH, a very talented PhD student from MIT, my laboratory, John Martin, thought of a very clever way of doing that. So what I want you to think about is a thought experiment. Not thought, actually, some of you might have done this experiment this morning. You have an orange, and you have, for breakfast, so you just cut it open in the morning. Now what happens?
the juice oozes out.
Nothing, right? It's like a installation in modern art museum, right? You just watch this, you say, it's cool, man, and then you go to the next one, right? Okay? Now you do the same experiment, now with a tumor, which is what John did. Now what happens? Do you see anything? Let's see the movie again.
It's falling
It
apart.
is oozing out.
Yeah.
Oozes out.
Is it oozing out? It's opening up like a lotus flower almost, right? Yeah. Now this is the advantage of being an engineer. So when this goes like this, using mathematics, standard mechanics principles, you can calculate how much force the tumor is generating. Right? And we did that. First in mice and then in patients. Tumors. And pancreatic cancer opens up the most. And among the breast cancer, triple negative breast cancer opens up the most, because that has the most matrix in it. So, the question is, how do you fix it? How do you get rid of that? So nearly 20 years of work, I will not go into how, they're very interesting treatments we developed and failed. We came up with a very nice solution, and this was led by two of my PhD students, Rakesh, a PhD student from Harvard, and John Martin, a PhD student from MIT. Really brilliant postdoc, graduate students. And what they showed was that that the widely prescribed drug for controlling blood pressure, antihypertensive drug, known as losartan, that costs less than $1 per day, more like 14 cents a day. What that does is, I'll not go into the biochemistry of it. What it does, it reduces two different components of the matrix. One is known as collagen, and the other one is known as hyaluronic acid. These are the two which are abundant in tumors, which are highly resistant to treatment. And he could reduce it. And when he did that, so let me just show you what some of the images from his work, PhD work. And what you see on the left-hand side, the blue color represents the matrix, imaged in a live animal. And the blue color and the green color represents the blood vessels, which are flowing, functional. And you see, you don't see too much green inside where the blue is. That tells you the matrix is compressing those vessels, that's why there's no flow. Now you give losartan, a week later, you see the blue color goes down and the green color comes up. Because there were vessels earlier there, but they just were not functional because they were squished. All right, so then when he did that, he could deliver more drugs. Both chemotherapy and immunotherapy worked better. So then we decided to test this idea in pancreatic cancer in collaboration with our colleagues at Mass General Hospital. And I'll not go into the details of the therapy, but the standard of care, so there are 3 types of stages of pancreatic cancer. One is when it's metastatic, this is advanced, that means just, then survival rate goes to about 12%, 5-year survival. But when it's localized completely, so that surgeon can move it, then it goes up to about 48%. All right? So we said, okay, let's take the locally advanced tumors, and the standard of care of Mass General Hospital is drug, chemotherapy called FFX, and radiation. And only about 28% of the tumors can be resected, become resectable. But when we added losartan, the number more than doubled. Simple drug, costs $15 for the whole treatment. All right? And what is really remarkable, this is not good enough, but for what is really good about it, that most of these resections were R0. You're not pathologist, R0 means there are no cancer cells left behind. So this was just unprecedented, and this was called a breakthrough in that year when it was published. So anyway, and now we are following up on that, and some other studies of losartan. The problem is we cannot get money for clinical trials for losartan. The reason is very simple. Losartan is an off-patent drug, so pharma company has no interest in. So that's why this work is not moving forward. So if you know of any donors, any foundations, and are there a lot of billionaires from IIT Kanpur, IITs, if you know any, please ask them to support a clinical trial. Because this would help not only people in rich countries, but even more in poor countries, who cannot afford $100,000 drugs.
So question for you, on the previous slide,
Yeah.
what's the difference between R0 and RT?
Oh, RT means radiation therapy.
Okay,
Yeah, that's an abbreviation.
okay, you got R0 there as well.
Yeah, R0 means no cancer cells left behind after resection. Yeah. Oh my God, lots of, yeah. Keep in mind, you're not going to have, yeah, go ahead.
So this is great. I'm wondering that if somebody is taking the anti-VEGF therapies like Avastin, which is intended to block angiogenesis, would that actually make the tumor worse and more prone to metastasis?
Hang in there for 5 more minutes.
Okay.
Yeah. Go ahead.
I have questions
Yes.
I saw your slide in which you were saying about dissecting about the lemon and the tumor also.
Oh no, it was an orange. Yeah.
Okay, sorry.
Yeah, all right.
I'm just curious to know, same things happened in the blood vessel also, and in soft tissue when we are dissecting surgery, it will have residual stress built up mathematically if you want to understand.
Yeah.
So is that generalized in all soft tissue, or is just in the tumor cell you noted it happens in the tumor cell after dissecting the residual stress was generated?
The residual stress goes down. Yeah.
Yeah.
Okay. That's it. Yeah.
So this thing is, you said the clinical trials were in pancreatic cancer. I assume it can be extended to other cancers?
No, we would like to do the next
That's
trial in
okay,
pancreatic
yes.
and other cancer, of course.
Okay.
So about 25% of the human tumors are what's known as desmoplastic. That means they have a lot of matrix. Pancreatic cancer at the top. Next is triple negative breast cancer. That also is very poor prognosis. And so these, and about 25% of lung cancer. So there are many cancers that will benefit from that. Yes.
I'm curious to know if there was any difference between the squamous and the non-squamous malignancies in vis-a-vis the institutional fluid pressure, and then the effect of losartan on them.
So I'm not talking about the fluid pressure.
No, in terms of
I'm
the
talking about
matrix,
solid pressure.
the whole matrix, yeah.
Yeah, I'm talking about the solid stress, not the fluid stress.
Right, yeah.
Yeah, so I'm about to talk about fluid in a
Okay,
minute.
okay.
Yeah.
So this slightly more general question is, when you do this, right, you provide losartan, reduce the pressure on the matrix and the blood vessel grows more, do you see that evolution of the cancer cells themselves to adapt to the changing, they have their own life force, right? Do you see their biochemistry modifying itself to deal with that?
They will get now more oxygen, they will change, yeah. they'll become more sensitive to many therapies, such as radiation, that requires oxygen. All right? So far I've discussed one mechanism why blood flow will stop, because somebody compressed the vessel, right? What else could be the other possibility?
Debris.
It got siphoned off somewhere.
Debris, hole somewhere.
Hole somewhere, yeah?
Debris getting in there. I don't know.
Okay, so hole, right? Leakage. So the molecule that makes leakage in our body is known as vascular endothelial growth factor. It was originally discovered at Harvard by our friend, our colleague, Dr. Hal Dvorak, under a different name, as vascular permeability factor. Because as I said, tumor vessels are very leaky, and he isolated this factor. He referred to it as VPF. But then Genentech did the more genetic work of that, and they referred to it as vascular endothelial factor. So it has two functions, right? So if you put VEGF on a vessel, it becomes leaky, right? And as I said to you, the tumors have lots of VEGF, because VEGF usually goes up due to hypoxia and many other reasons. So now, if you have a lot of VEGF, this is what happens in tumors. So what we proposed was not to completely wipe it out, but reduce it. So the leakiness will go down. The reason we don't want to completely wipe it off, remove it, because VEGF is known as endothelial growth factor, is also required to maintain the endothelial cell integrity. Without it, the endothelial cell will die. So companies like Genentech and many others were developing drugs against this to starve the tumor, to cut off the blood supply, based on the work of late Dr. Folkman at Harvard, who in 1971 proposed the best way to kill cancer is a very surgeon's thinking, is to, well, what's the best way to cut a tumor supply? Take the tumor out from the body, right? There's no blood supply. So he said if you could do this in situ while the tumor, and he suggested, why don't you give a therapy that will kill tumor vessels? Now here is the problem. If you completely cut off the blood supply, like the surgeons do, that'll work like charm. The problem is, if you don't do that, then the tumor becomes angry. It begins to metastasize. You remember I said to you in the picture that areas which are hypoxic, the regions below, they are the more metastatic. So you're making now whole tumor, or larger regions of the tumor, more metastatic. And that's exactly what was going on when they did that trial, and did that very first trial. So then they decided to add chemotherapy to it. And that worked. So I said, okay, I went to this meeting and I said, it was about 8,000 people in the room. I said, well, could you explain to me, by cutting off the blood supply, how are you going to help chemotherapy? You wouldn't be able to get there. There was silence and people sort of looked at me, what's wrong? So I said, is it possible that what you're doing is by, in patients who are benefiting, you're actually repairing the blood vessels with this drug? And they looked at me like I had two heads. And they asked me, what is Dr. Jain smoking? And I still like to know what I'm smoking. So that's when I proposed this very counterintuitive idea. Let's use the drugs that were originally designed to kill the blood vessels to actually improve them by using the proper doses. And that's what we refer to as vascular normalization. The idea is that in normal tissue, the molecule which form vessels and that kill vessels are beautifully balanced. But in tumors, this balance tips in the size of pro-angiogenesis, that means there are a lot of blood vessel formation. Except these vessels I have normal, as I showed you. And what we proposed was that why don't we bring the imbalance to balance? And if you could do that, then we would have not perfect vessels, but closer to normal. And the word we coined for what that was normalized. So again, you can imagine this concept was not well received. So I had trouble getting funding for many years. But finally he said, okay, well, let's present the data. So we did this work in mice. And then he said the breast cancer is going in breast, and if you give this therapy the right concentration, the vessels get straighter. In the beginning they all screwed up, but then you see they get straighter, narrower, or begin to look like normal vessels. So this is what we refer to as normal. When that happens, blood flow goes up, oxygenation goes up, more drug delivered, tumors respond, etc. Again, same issue. When you publish this work, it's Mickey Mouse. So, very fortunate that being where I am, that we were able to do our 40 clinical trials in different tumor types, different anti-VEGF drugs. So I'll not go into the details, but the bottom, let me just give you one example, all right? So this is a newly diagnosed brain tumor patients. Their usual survival is about 14 months. And so when we gave these drugs, oral drug, to 40 patients, this trial was led by Dr. Tracy Bachelor, my collaborator. In 20 patients, the blood flow went up in the tumor. 10, it remained same, 10, it went down. So he came to this lecture prior to my normalization hypothesis. And I asked you, okay, so which patient will live longer? You would say these guys because the blood was starving, right? But if you're listening to my talk so far, you said these guys will live longer. And indeed, they had a survival advantage of 9 months. And now we have seen this in many other tumors, and others have seen it. So these are from our trials. So, and we're finishing this work, and this is now well accepted. The immunotherapy was just coming up, all right? And it wasn't working in many patients. So we asked the same question. What happens if you have to repair the blood vessels? Would immunotherapy work better? And indeed, this is the work of a postdoctoral fellow published 23 years ago, 13 years ago now. And again, he showed that when we do this, I will not go into all the details of it. You can see the blood flow goes up, and hypoxia goes down, and the survival of tumors, mice is doubled. All right, so this was cool. But what is nice is hundreds of laboratories have reproduced this. But what is even really gratifying is that FDA has approved 7 combinations of this approach. All right, and currently there are about a couple of hundred trials testing this worldwide. And the number, this is number from 2020, probably the numbers of trials have gone up. But to me, even more gratifying is that the pharmaceutical company has changed their thinking, and now they're developing new sets of antibodies that instead of giving 2 different drugs, anti-VEGF and checkpoint blockers, now they're making antibodies that target both. See that? So you can bring immune therapy and at the same time normalize vessels. And in China, they just finished last month a randomized phase 3 trial, and they got their first approval. So right now, this is going to be the new wave of drugs. And it's so, I cannot tell you how gratifying it is for a scientist to watch the whole drug strategy change because of the work proposed many years ago. So as you know, do you know what the hottest cancer therapy is right now? Have you heard of CAR T therapy? All right, where you take the immune cells from the patient, re-engineer them, right? Genetically engineer them, put them back, they find cancer and kill them. It works in leukemia, it changed the paradigm. But it doesn't work in solid tumors. Same problem. We asked the question, is it because the blood vessels are abnormal? And the answer is yes. So we said, okay, what if we normalize blood vessels? And using anti-VEGF therapy, and indeed, as shown here, you can see that if you don't normalize blood vessels, the red color are these CAR T cells. In this brain tumor, you don't see any. But here you see some red guys. See the red. These are the CAR T cells. They were just absent completely. And that's why this therapy, when you give systemically, doesn't work. And when you just do this, I'm not going to the detail, there are more CAR T cells, and they are more active. And the animals survive longer, 2 different models. So, inspired by this work, again, this is just what I just said to you just now. Now what we're doing is making CAR T cells that make anti-VEGF therapy themselves. All right? So now, similar to the antibody that does do both, now we're making CAR T cells that do both, and hopefully this will move forward. So we are very excited about that. Just want to end very quickly. I know we've gone way over time, but this will give you less time for quiz. So you can look at it in a positive way. I just want to quickly begin to end by saying that this whole concept of normalization can benefit other diseases besides cancer that afflict about 500 million people worldwide. I will not go into the team. I'll just give you 3 examples successful. One is that patients get these benign tumors in the ear around the auricular nerves. They lose their hearing. Most of these are young children, and some of them never heard anything. And what we showed, if you use like homeopathic dose of these drugs, very low dose of anti-VEGF therapy, and the hearing came back within a week because the vessels were abnormal, and that was causing this problem. And so, as a result of this, this becomes the standard of care. It's very gratifying. We found out that losartan can also do this. Not going to that, but tuberculosis, which afflicts about 1 billion people worldwide, have the same problem. All the NIH money and Gates Foundation money had been going into same way as cancer cell field. We're looking at cancer cell resistance rather than looking at microenvironment. We said, okay, does anybody look at the microenvironment of TB? No. But guess what? If you look at lesions in the lung, they look similar to the tumor I showed you earlier. And now, if you inject a drug, guess what? It only gets part of it in the margin, similar to what happened. So how are you going to cure? And that's why TB develops resistance after a while. So we showed that losartan, this paper just came out last year, that if you combine losartan with a anti-VEGF therapy, you can deliver 8-fold more bedaquiline. That's the most recently approved tuberculosis drug. So hopefully the Gates Foundation, supporting this work, will do a clinical trial in South Africa based on this. So let me just conclude my talk by saying what I hope I have conveyed to you. The tumors have abnormal vessels, and many other diseases too, and abnormal vessels may have trouble. If you want to see in a very simple layman's language, I wrote an article about this Scientific American in 2008. What I showed you is 2 different strategies, how we have taken them from bench to bedside. I just want to end by pointing out something new. This is a really new work from our laboratory, that moderate I'm sure you've been reading a lot of articles, exercise helps cancer patients, right? Helps a lot of things. And this is the work. And how does it help cancer treatment? What we found out is exercise normalizing blood vessels. And when that happens, you see that tumor response. So right now we are trying to figure out why it's normalizing vessels, and it's not so forth, but this is very exciting. The good news why exercise doesn't cost as much as drugs. So something you guys think about doing. But what is even really good is you don't have to do really heavy duty exercise. There's moderate exercise good enough. You don't need to do that. If you do no exercise, that's not good. If you do too much, it's not good either. So let's end me my presentation by acknowledging my current lab members and some colleagues at MIT and others. These are collaborators in glioblastoma brain tumors, collaborators in other area treatments. These are my collaborators in the tuberculosis project. For the last 39 years, I've been teaching a 4-day course, continuing medical education course on this topic, which I summarized to you in the last hour and a half. I know I saw one student here who took this course. Could you raise a hand again? You did. Can you please convince the rest of them to come to the course? All right. This course is this year in November, and it's in person. All right, I really hope you consider doing that. It's got very distinguished faculty, including Nobel laureates. But really, the people who are most important are the 250 graduate students and postdocs I've had the pleasure of working with in my career. Without them, none of this would be possible. And they are the reason, they are my extended family. They're the reasons why I cannot ever retire. Because how do you, one of these are brilliant people. And I wouldn't be where I am without the support of my family and my wonderful, beautiful wife sitting there who keeps me honest. and my twin daughters who will be seniors this year and be graduating in spring. So thank you very much and be happy to answer any questions you have.
Thank you so much.
You had a lot of time for Q&A.
We'll keep it short. since
I've taken so much of your time already.
Yeah, we did have intermediate questions already, so we'll try to spend another maybe 5 or 6 questions in total. So, because we do have an address on IIT Agni and the networking event. So, Dr. Jain, thank you so much, and please have a seat.
Oh no, it's okay.
You can have it. Okay, is everyone okay? Audible?
Yeah.
You can also use.
Yeah, I have this so you can hear me.
You can use your seat mics to ask questions in case you're in rows behind. So, all right, we already have a lot of questions. So, okay, maybe let's start.
Why don't you're the moderator, so you choose it.
All right, I can choose. Yeah, Rupesh, why don't you take that?
Yeah, so my question for you...
Before Africa, she takes bribes, okay?
I know her. So a question for you, right? So we all come from India, where cancer rates are skyrocketing because it's much more...
But that's part of development and living longer.
Sure. But if you look at most families...
pollution, and India is now the king in pollution.
It's incredible. So most of us who come from India, families in India, all have cancer patients now. A lot of us have cancer patients. My mother-in-law has now got lymphatic cancer. I believe it's because of the environment. Now the question is that your research, which actually is really opening up new directions, how much of it is been taken up by the medical community, research community in India? And now there's like serious investment that's going into healthcare, like the AIMS, for example. There are AIMS campuses everywhere. So there's real work that's being done, and now India is a much more richer country. So how much like IIT Madras now has a medical program, actually, believe it or not?
IIT Kanpur is starting a whole
Sure.
medical
All IITs have started.
Yeah, so the question I have for you is, how much are they talking to you, and how much are they using what you know, what your work and your breakthroughs?
I really don't know the answer to that question. I served on the Department of Biotechnology Advisory Committee, I don't know, 20 years ago. And it required going to India once a year, which I did for 5 years. And then I stopped, And at that time, they weren't doing anything I was asking them to, so that's when I quit, But I imagine things have changed in India now. I'm going to be very honest with you, I'm very disconnected with India right now. I should not be, but I am. And in terms of what's happening there or not. Not because I don't want to, it's just that I just don't have enough time. But I'm happy to do that. I know the IIT Kanpur folks have approached me, and I'll certainly help them once they get to certain stage. I do know that IIT Bombay has spun out very nice technology for making CAR T cells much cheaper than they are here.
Actually, they are working with hospital in Bombay.
Yeah, I know.
Tata Memorial? Is that Tata Memorial?
Yeah, but they're working with Tata.
It's in the works.
I know about that work. Yeah, it's very nice.
Super. I do see a lot of hands raised. So what we'll try to do is we'll try to club a few questions, if that's okay. So maybe we'll take the entire row after you.
So I'm going to switch. By the way, amazing research, and I think it's really helping, all the patients. I'm going to ask a little different questions about different, alternate therapies, like alternate therapies.
Yeah, alternate.
Like acupuncture.
Yeah.
About 15-20 years ago, I went to an acupuncturist in Boston, Chinatown, and there was a patient, cancer patient, a doctor, and his wife was also orthopedic surgeon and had come there. And he was given like 2 months, but then through his therapy, he extended, he was going on like 8-9 months already, right? And I was talking to the therapist, and he was saying that many times cancer patients come when it's terminal. They don't come like earlier in the stage where they could help. And they come at the terminal, so only thing is left is essentially make their quality of life better and extend that, few months. So do you know anything about that acupuncture or anything that's being used or in combination with these therapies?
No, I don't do any work in that area. I really don't. So I cannot give you a knowledgeable answer. But I know that there's a lot of work going on in China on that. And actually with the NIH funding being cut in this country by the current administration, I think it's just a matter of time that China will surpass US in medical research. Right now, US is so far ahead of every country. We have gotten.
Any thoughts along yoga then?
It's what?
Yoga? Helping cancer?
Again, I don't know any of this. Yeah, I don't have enough time to do this.
We try to limit our questions now quickly, and I'll try and club a few questions, so yeah, please go ahead.
So you've talked about these specific environments for cancer cells, like hypoxia, low pH. So my question is, how deterministic are these markers? So if you were going to clump 3 or 4 of these observations together and give it to a computer model, just based on those observations.
Yeah, AI, so people have done that.
How accurately can it diagnose?
So actually, they are the best predictor for radiation outcome. A very nice study published on that already. But it just, AI is not checking all these things, it's going to be flood of papers. Because that allows us to do that. So you can, but the hypoxia is the best predictor of worse radiation therapy. I think there are going to be similar papers coming out for immune therapy, and just a matter of time.
Okay.
We had a question from this side next.
Thank you so much, Professor. My question is building on the same one, is how best we can combine mathematical modeling and AI-driven image analysis of malignancies to see how optimal the drug delivery has been and what the results are.
So do you have your access to internet?
I do.
No, right now. Do you have your iPhone?
Okay. You want me to do something about it?
Yeah, I just want you to look at Nature Reviews Cancer. This is like the leading #1 journal.
Yes.
And look at Jain, Jain, and then put 2025.
Oh wow, this is as recent as this. Lovely. Yeah.
What do you see?
We have a lot of articles here.
No, 2025, I have only one article in Nature Reviews.
So the fibroinflammatory response to cancer?
2025, Rakesh K. Jain, Nature Reviews Cancer.
This is what has been pulled up.
Maturation of cancer?
And this,
Oh, using mathematical modeling and AI to...
You got it. This guy got it. Okay, so can you read the title to these guys?
Yeah, using mathematical modeling and AI to improve delivery and efficacy of therapies in cancer.
So please take a look at that article. Just came out.
okay.
Just this very topic you're talking about.
Right. Yeah, I will go through it. Thank you so much.
I think those of you who are mathematically inclined will really like this article because it pulls together the work of 50 years and brings it with AI, how radiomics and all that are.
I know you've been raising a hand, but yeah, please go ahead.
I was telling my wife on the way here that if I were to start a career again, I would be doing computational biology, and you have just reinforced that, which is. So my question was.
Take a look at that review article.
I intend. For vascular normalization, right? You mentioned losartan and VEGF.
Are there other research kind of tracks, investigating ways in which normalization can happen?
Yeah, so I can refer you to another review article of mine, but there are 18 pathways we are working on, others are working on. Exercise, the most recent one.
Okay.
Yeah, we had one question there.
If you just look at my publication, just look for the review on vessel normalization. Yeah.
So sir, a different question, like for detection, how, because now you know that the vessels are not normalized, is that something that can also be used for detecting the cancer, or is it too invasive?
Well, you want to, incidentally, blood vessels begin to become abnormal at the very early onset in pre-neoplasia. Not even before cancer cells have become fully neoplastic, cancerous. There's a stage they go through called pre-neoplastic stage. That's when the blood vessels begin to become abnormal. And you know why? What do you think cancer cells are most afraid of?
Blood supply?
No, they want blood supply. What are they most afraid of?
Blood supply.
Immune cells.
Immune cells.
Immune cells.
So if the vessels how do they think you're going to defend yourself? Right? So they become, this will be published 20 years ago, that during pre-neoplasia, the vessels become abnormal. So we have a way of looking at, in a mark, different stages of cancer, pre-neoplasia, neoplasia, hyperplasia, the whole thing. I don't want to go into biology of it.
But that is invasive, right?
No. Just in the beginning stages, yeah. So the beauty is with the radiomics, going back to your question, they will begin to combine that. So the AI will help with that. Has not come in yet because it hasn't sunk into them yet, but I'm thinking about writing an editorial about it. Yeah.
So can I also follow up, sir?
Yeah.
So.
No, you have to ask her permission.
Yeah, sorry. Can I follow quick?
Very quickly,
Yeah,
then we have the last two questions.
So this is a dumb question, but when we normalize the blood flow, right? And since you said cancer cells do like blood, so isn't there a risk that you are making them more stronger than where they were when the blood vessels were not normalized?
So you don't do this in the absence of any drug.
Oh, okay. So it's like got it.
But independent of drugs sometimes, just immune cell begin to work better. So many times, not always, you don't want to take a risk, but many times, this normalization of vessels alone can control tumor, but I wouldn't do that. The reason it can, because in those cases, immune cells come in and they begin to do the job.
Yeah.
So maybe it's related to what she was asking. So just like in HIV, at-risk patients have been given the drugs in a preventative manner.
Sure.
Would giving losartan in a preventative manner to the high-risk patients prevent the vasculature from becoming abnormal?
Well, we don't know that, but we did a retrospective study that has been published from Taiwan where they had very careful record of drugs. They found out the patients who were taking antihypertensive drugs, they had a better, lower incidence of cancer.
Okay, interesting.
Yeah. But again, it's not a causative study. It's just retrospective studies are just speculative, right? Yeah.
So high blood pressure is good then.
Huh?
High blood pressure is good.
No, high blood pressure is not good at all.
Because then it will make you take that drug.
It's better not to have high blood pressure. Yeah.
I have this question.
But you can take losartan at lower doses without control changing your blood pressure. Yeah.
So similar question I had is like if somebody is...
So then that pancreatic trial, we gave a dose which doesn't change blood pressure.
Similar question I have, like a patient with a low BP,
yeah.
If they put on losartan...
We cannot give them that. Yeah. So when we did the trial, those patients were excluded.
Okay.
You had a question.
Yeah. So I'm a postdoctoral fellow in Kemi Leissure's lab at Mass General Hospital, and currently we moved to Tufts School of Medicine. So my work is basically engineering the bacteria for therapeutic drug delivery inside the tumor tissue and inside prevent gut infections and all. So some of the bacteria like E. coli initial, I work with another commensal which is E. coli.
So you working with E. coli?
Yeah, yeah.
Okay.
So mostly the commensal E. coli which are good for human gut. So so E. coli initial, which is FDA approved and it's known for anti-inflammation and all that. So when I work with these bacteria, I see that in tumor, so initial or E. coli HS, they have some inherent anti-tumor activity. Like they regress tumor, they do not let it grow. I'm engineering to make them, you know, a very good vehicle to deliver more therapeutic there. So I was wondering, do you think that these bacteria or anti-tumor bacteria, they have some sort of relation with this vascular tumor normalization or things, or they impact the vasculature inside the tumor? So is there any relation between anti-tumor bacteria and this
So since you work in this area,
yeah.
you must know a person named Neil Forbes.
Yeah.
You know him?
Yeah.
Okay. He was my postdoc. Yeah. He did the very first study ever published on how bacteria get into the tumor. It was published in PNAS.
Yeah. It goes in.
You saw that paper? Yeah. So that's Neil Forbes, yeah. So, and he did the imaging of these into the tumors. And they went into hypoxic region.
Okay. Yeah. He's currently in Canada. Sorry.
Last time he was at UMass or someplace in the middle of this country.
Okay.
Where he moved to Canada. Yeah.
Yeah, I've read his tumor, Nature paper.
No, he's one of the leaders. Yeah.
Yeah.
I think with that we have maybe just one more question. Okay, yeah, please go ahead.
I just was a little confused on, so you proposed combining losartan and with the anti-vector therapy, and then you also proposed putting putting this therapy in CAR T cells. So I just wanted to know, I guess, what the implications are for both of these, because I know CAR T is like so up and coming right now. So I wanted to know like what your thoughts were, and like, is it organ specific or?
I didn't quite understand your question. Well, your question is, can you put, we are putting VEGF in CAR T cells.
Yeah.
So you're asking, is that organ specific or anything?
Like, yeah, and then combined with, or I guess you also proposed, if I didn't get it wrong, putting the losartan.
No, we are not putting losartan in CAR T cells.
Oh, non-CAR T cells, but combining it with the anti-VEGF therapy.
Yeah. So we have done that in tuberculosis.
Okay, so that's specific to tuberculosis.
Well, that's the only thing we have done.
Oh, okay.
It's not that it's specific. It's just you can do only so much. But after you're done with your current study, you can come to my lab and work. See that opening?
I'm an undergrad, so.
Well, you would be graduating or something. Yeah.
Thank you.
It's okay.
Yeah, absolutely. Next, last question.
I have a question. Given, I think you referred to it, but what do you see as the future of a lot of this research given the current environment with respect to political funding?
I think all the research will shut down.
To that extent, it will shut down.
Well,
Not
look
slow
at, they have
down.
taken away $400 million from Columbia University, right?
NIH grants are going down.
They took out $400 million worth of NIH grants from Columbia University, right? You know that.
Yes.
It's just a matter of time. Some other university will suffer. So, yeah, the future is bleak right now, but my hope is that it's transient.
And are you seeing private, as a follow-up, private foundations are willing to step in or not yet? I mean, NIH is huge, obviously.
Nobody can make up
That's
for it.
it.
I mean, look, they are the world's largest research funder for. That's why I came to give this talk, so you guys would donate money. What? Do you think I would accept you to give this talk?
Okay, so
I know Indians are very rich. I know that.
How much do you influence?
They have the highest per capita income in this country. So that's why I came here. And IITians are the best. Yes.
We need to get you to Silicon Valley.
Everybody has
Dr. Jain, I have the same expectation that you are giving money. I run a lab.
Every event and every action is quid pro quo, right? So one of Newton's laws. So I think
I know somebody from biotech companies or pharma, then you guys can convince them how to. So yeah.
So regarding funding, at what
incidentally, no matter what happens, I'm not retiring. I love this profession.
What stage it is in?
Huh?
What stage? Is it like real pure research, or there's some drugs have been developed, or what stage is it? Because depending on that, funding may differ, and we might get
What are you talking about? What stage is what? I described to you what different things are.
Right, but how close is it for, it's in the trials right now, or trials have been succeeded? Maybe we should talk more details.
Happy to, once somebody offers money, I'll be happy to talk.
Absolutely. So I think we can definitely take a lot of further discussions and questions offline as well, and take it forward from here.
Well, thank you very much for your...
Absolutely, Dr. Jain, it's been wonderful. Before we move on, I would like to request Durya to give a small token of appreciation from our end to Dr. Jain for sparing his time on a Saturday.
Thank you
and
so
coming
much,
in, yeah,
Dr.
and
Jain.
I'm like you.
Ever since I saw your talk at Pan IIT, and I approached you after that, it's been on my list to have you come and talk to IIT group. And finally, Soumya, thanks to Soumya to help organize this.
Thank you
Apologies,
so much for
you had to adjust your ride and all of that today
But
morning.
you know what? This bouquet doesn't belong to me, it belongs to my wife. So
I will
could you
give it
please
to
come
her.
here?
Yes,
Because
please.
everything I have done
Jackie,
wouldn't be possible.
Absolutely.
where-
So we would
Jackie,
reach out to her. Well,
Please come and get the bouquet. Yeah, for you.
just such a small thing. But thank you, Dr. Jain. I don't know much about biology at all.
I don't
but,
either, I'm blind.
but I think what was really fascinating is to hear you talk about the intersections of the discipline and how the intersections bring novel approaches. And that's sometimes, I think all of us, it's good to remember all of us, is that different opinions coming from different perspectives actually enriches the science in the end. So, thank you so much. Take care of him, take care of yourself, being part of this community.
Thank you so much. Thank you so much to Jackie. Thank you so much, Dr. Jain. I hope we've all enjoyed, as an engineer or a techno brain, delving into medicine. So thank you so much for your time. I'll hand it over to you, Durya.
Sure.
To give your input.
So just wanted to wrap up a little bit again. Thanks to Dr. Jain, thanks to all of you for taking the time to come and listen to this. It was really good to see so much active participation. Hopefully Dr. Jain didn't mind all the questions along the way. So I just wanted to say, we hope to continue bringing in more talks. I don't think we'll be able to match Dr. Jain's expertise in that sense, but we hope to see you folks more in a lot of other sessions. For the next month in April, actually, we have 3 events coming up. The one that I want to call out very specifically is on April 26th. We will be at Brown University. Ganesh, who's a VP of IT Agni, is coordinating that, and it'll be on the blue economy. We have a really excellent black panel of speakers, so we hope to see you out there. And before that, earlier in Jan, we have a workshop. So IT Agni's mission is to really promote STEM awareness, STEM education, STEM-related activities in this area. That's where we are all passionate about. And so we have a 3D printing workshop for kids coming up. It's also a combined fundraiser for Vision Aid. And then we have a social networking event coming up. So you can find out about all these events at iitagni.org. That's the best place. It's very easy to remember, best place to go. and we hope to see you. And then today, normally IIT Agni events, we have a signature in all our events. We have samosas, thanks to Rakesh. He's been bringing samosas to every IIT Agni talk for quite a few years now, and chai. But because HKS, we couldn't get food, so we are hoping that you folks can join us at.
Pleased to meet you. And sir, you?
I'm Ganesh. I'm from IIT Bombay.
Okay.
I went to high school in Kanpur, so I actually grew up for several years in Kanpur.
Oh my goodness, yes. I didn't grow up in Kanpur. I just went to IIT Kanpur. Yeah. Yes, sir. You?
I'm Anil Sehgal from IIT Bombay. I'm a professor at Tufts University.
Oh, okay.
I'm Shailesh, IIT Mumbai.
Hi, pleased to meet you. Are you thinking about going to IIT also?
I'm going to MIT.
Oh, not quite as good, but it'll do, Yeah. And you?
I'm Durga. I'm not from IIT, I'm from REC.
Oh, okay.